Hi there, and welcome to 2.10, which is all about enzymes. So, first of all, what is an enzyme? An enzyme is a protein. Make sure you know what enzymes are made of. So, enzymes are a type of protein, which is why they follow proteins in the specification. So, they're made of protein, and more specifically, they are globular proteins. Okay, so they're kind of spherical shape, roughly. Now, enzymes are very, very specific, which is really, really important. Each enzyme has a different active site. So, in a kind of a crude way of drawing it, we could draw an enzyme with an active site like that, and an enzyme with an active site like that, and an enzyme with an active site like, yeah. So, when we then get our substrates, which might look like this, they only fit into one type of enzyme. So enzymes only work on one type of substrate, and that's because of the 3D shape. Okay, so they're specific in that their active site is only complementary to one substrate. Okay, and make sure you really understand what specificity means, and make sure you always use the word complementary, not matching or same or anything else. Always the word complementary is really, really important. Now, the way that we used to teach that enzymes worked was something called the lock and key model. Now, the lock and key model was very much like how I've drawn it just there, in that the active site of the enzyme had a specific shape and it was rigid in that shape, just like the lock is on your house door. And then the substrate is an exact match, it is exactly complementary to the shape of that active site, and it binds. And when it's a very complementary shape, it fits perfectly and it catalyzes the reaction. Okay, but the key point about the lock and key model is the active site is a fixed shape that binds to the substrate. So we need to know, first of all, that the active site is what we're referring to as the lock, and the substrate is what we're referring to as the key. And we need to be specific that in the lock and key model, the active site, the lock part of it, is a fixed shape that can't change. Now, sometimes they'll ask you to label diagrams to do with this. So let's just make sure you're really clear about how you would label them. So you would start off with an enzyme and a substrate. Then when the substrate binds into the enzyme, you call that an enzyme substrate complex. And then once it's catalyzed the reaction, the enzyme stays unchanged, but the substrate has changed. So in that case, let's say it's broken into two products. Okay, so make sure you can do the labeling of that. So we've got enzyme and we've got a substrate. Then here we've got an enzyme substrate complex. And at the end, we've got our enzyme, which remains unchanged, ready to be used again. And we've got our products. Okay, so just make sure you can easily draw and label a diagram like that. Now, the next part is that through scientific experiments, scientists actually realised that the lock and key model wasn't really that accurate because, in actual fact, the shape of the active site is not rigid. It can actually change. Okay, so we came up with a new theory which is called the induced fit model. And in the induced fit model, the idea is that it's more like a glove and a hand. So the analogy for this is that the glove is like the active site of the enzyme, and the hand is like the substrate of the substrate that's going to react. And the idea is that the active site shape adjusts slightly to fit the substrate. Okay, so your hand and the glove are nearly complementary shapes, but not exactly. On the lock and key, they were exactly complementary. On the induced fit, they're nearly complementary, and then the active site shape adjusts slightly to fit the substrate. It is actually a little bit flexible. Okay. The next part is these very familiar looking graphs about activation energy. So the graphs look like this. Well, we've got the energy at the start of the reaction here. Then we need some activation energy to start the reaction off. Then as the reaction progresses, the energy goes down. And then we've got the energy of the product. So make sure you can label a diagram of this type first of all. So here, this line here at the start is the energy of the reactants, the start. And then here at the end, this is the energy of the products. And most importantly is the height between the top here and the reactants. And that there is called the activation energy. So they might give you a graph and ask you to label it. They might give you a graph and ask you to say what the value of the activation energy is by using the numbers on the axes, something like that. So this is the activation energy. And activation energy is the amount of energy that's needed to start the reaction. So let's write that down. So the activation energy is the energy required to start the reaction. And from a graph, it's always going to be from that start point, the energy of the reactants, up to the peak of the graph, up to the top. All right. Now, if we were to add an enzyme to this reaction, we would actually reduce the activation energy, which is why enzymes are so useful. So enzymes reduce the activation energy. And very often, what they ask you to do is they draw a graph, something like the one we just saw. So they'll draw a graph something like this, and they'll ask you to draw on the line that shows what would happen if you used an enzyme. And you need to draw a graph that starts at the same level, goes up but not as high, and ends at the same level. And that's really important, okay? I'm going to just draw that again a little bit neater, and just to show you again. So let's spread it out a bit. So up and down. So that black line is without the enzyme. Oops, can't spell. And you can see, I really can't spell. And you can see that the activation energy is from here to here. And then with the enzyme might be the bit they ask you to draw on. Make sure you start at the same point, but just don't go as high, and then join on at the end. Oops, end at the same point as well. Be really careful, use a pencil so you can rub out if needed. They must start at the same point, they must end at the same point. And you can see that now our activation energy has been reduced. So the red line is with the enzyme. So chances are you're going to have to draw one of those lines. So be really careful that they start and end at the same point. And the enzyme one is lower, the peak is lower. The reasons why they lower the activation energy, let's put this in here. So enzymes reduce the activation energy by, first of all, it helps bonds to form by holding the enzyme and the substrate close together. And secondly, it helps bonds to break. Probably should be the other way around. You should probably break bonds before you make them. But anyway, helps bonds to break by putting strain on the existing bonds in the substrate. Smell my spellings. Okay, so it puts a strain on the bonds and helps them to break more easily, and then it just holds them in the right place, just holding them just in the right place to form the new bonds that are needed to make the new products. Now, enzymes have different names depending on where we find them. So we get intracellular enzymes that are inside cells, and then we get extracellular enzymes for enzymes that are found outside cells. Okay, so intracellular enzymes are enzymes that are inside cells, and extracellular enzymes are enzymes that are found outside cells. Now, obviously, the enzymes need to be made inside the cell, and sometimes they're released by exocytosis to be extracellular, and sometimes they stay inside to be intracellular enzymes. And we need both. Okay, both are really, really important. Let me just double check that that's everything. Yeah, that's it. So well done. Another spec point done.